All right.
Do you need?
No, I always do this a little late.
Oh, okay.
So, let's see where. Stephanie said she'd bring them down. Are they right outside? The treats. QL, can you run up and see if Stephanie has the cart of treats? I don't know. Well, what we could do is we could start like 2 minutes late so everybody can run and grab a treat really quick. We'll pass it through the rows.
It's probably a shorter talk, so it would be.
It's totally okay.
Yeah.
And Janie's in my class. And Adam Van Dellen that just sat down in that back corner. Okay, we'll start. Welcome, everybody. Thanks for being here today. Oh my goodness. We got a Hope grad who graduated just a couple years ago, 2016, and she's back. And we're so excited to have her. She graduated in physics, but she almost also graduated in dance. She was almost effectively a full dance major, but she didn't quite get enough classes there.
Yeah.
She did research with the accelerator, nuclear forensic stuff like FBI, CIA stuff. Unfortunately, the project didn't work.
Nope.
In the end, auto paint was too complicated. Come on in. Welcome. And she went on from Hope College and went to Northern Illinois University. She got a PhD in nuclear physics with an emphasis on radiation therapy. And so her postdoc was on radiation therapy. And then she moved on and doing a postdoc with MRI-guided online adaptive radiotherapy.
Yeah.
That's a mouthful.
It's a lot of words.
What does that mean exactly? Well, she's going to tell us about it. She's going to tell us about it. So thank you so much for being here. And the show is yours.
Great. Thank you so much. Thank you for the invitation and for the introduction. So we're going to talk about a deep learning-based automatic contour refinement method for MR-guided online adaptive radiotherapy, which, as Andrew said, is a lot of words, but we're going to go through all of it. So for our roadmap today, we're going to start talking about radiation therapy. I'll talk about, kind of give you a brief overview of it. We'll talk about how MR-guidance can improve that process, and then how online adaptation and online adaptive radiation therapy further improves that. Then we're going to take a little detour into deep learning, give a brief introduction there, and talk about how it can improve medicine. And then we'll come back and talk about the research that I'm actually doing on this automatic contour refinement workflow. So radiation therapy, as defined by the National Cancer Institute, is a form of cancer treatment that uses high doses of radiation to kill cancerous cells and shrink tumors. We use many different forms of radiation in this process. The most common is X-rays in the megavoltage range, and that you can find pretty much anywhere. Any major hospital will have radiation therapy with photons. And then less common, you have to go to maybe an academic center or like bigger healthcare systems will have proton therapy or electron therapy. Also, we can use radioisotopes in various different forms. And then internationally, you can also do carbon ion therapy or helium ion therapy. There is some research in the United States with that, but it's still a long ways out. But they're internationally in Europe and Asia, you have those as well. As I go through this talk, you'll also hear me refer to dose. And in that case, with radiation, we're talking about the amount of energy deposited per unit mass. So dose, the units we use are gray, which in SI units is a joule per kilogram. So the workflow that a patient will go through when the patient is told that they're going to get radiation therapy, they'll come in and first get what we call a CT simulation. So this is a CT where the patient lays in the orientation that they would receive the therapy in. And then the patient leaves for a little while, and the 3D image that we got from the CT goes into the treatment planning process. The first thing we do with that is contour or segment the tumor, as well as all the organs at risk, or OARs. And these are used to calculate how much dose is going to be delivered to that tumor or an organ. And then it'll go into a treatment planning optimization process where we simulate how the radiation will penetrate through the body and try and get the lowest dose to the healthy tissues and the highest dose to the tumor. And then finally, the patient will come back and receive their treatment. A little bit more about the contouring and treatment planning process. So as I mentioned, the treatment plan is a simulation of how the radiation passes through the patient. So the CT has a bunch of densities in it that models the patient anatomy. The radiation, the x-rays will be using Monte Carlo simulation to look at how that radiation will actually attenuate as it passes through the body. And then that will calculate a dose in each pixel of the CT image. The contours assign a pixel to an organ, and then we can using summation, we can just summate the amount of dose in each of the organs. And that's how we can tell how much dose that we're giving to an organ or the tumor. And the goal of the optimization and the optimizer is to maximize the dose to the tumor and minimize the dose to all the healthy tissues. So you can see in this image, let me get my pointer going. There we go. You see a tumor volume here in bright red. This is a high dose, and we're able to get a pretty conformal dose distribution there. And we can minimize the dose to all the healthy tissues, such as the kidneys, which are outlined in orange. And then you can see the spinal cord here is a light blue, which represents a lower dose. And generally, the radiation oncology team is given about 1 to 2 weeks to complete this process. And they work on many different patients during that time. Then for the actual treatment, the patients come in for daily treatments, and they can last anywhere from 4 to 8 weeks on average. And that just depends on the tumor type, the cancer type, where the tumor is located. Cancer is obviously a very big field, so it just kind of depends on what the actual disease is in that case. So this is one of our treatment rooms at MCW at the Medical College of Wisconsin. So this is your patient table. The patient lays on here, the feet go on this end, and the head is over here usually, and they can lay on their back or on their stomach. And this table has 3 translational degrees of freedom, so up, down, side to side, and in and out, and then it can also rotate. This here is our linear accelerator, or our LINAC. The radiation comes out of the head here, comes down into the patient. And then we also have these onboard imaging systems, which can do radiographs, which is an X-ray that you would get if you broke your arm, or it can rotate and get a CT. And this whole thing is on what we call a gantry, and it rotates 360 degrees, so you can get a bunch of different beam angles there as well. Now, one of the problems that can occur because these treatments are so long is that the anatomy in the patient can change from day to day. So one of the things that is a benefit could be that the tumor is shrinking, which is really good. But also can cause problems because we simulated how the radiation was going to pass through, and now if that anatomy is changing, our simulation is no longer correct or no longer accurate. And so the tumor shrinking can happen. You could have the bladder, it could be more or less full. The weight changes, so they could have gained or lost weight, especially if they're going through chemotherapy at the same time. And also any sort of bloating or air pockets that occur throughout the GI tract is a big issue as well. And so what can happen is because our model of the patient changed, the simulation is no longer correct, and we could be giving more dose to the healthy tissues or less dose to the tumor. And both of those can cause some serious side effects. So this here is from a paper looking at how these changes in anatomy can affect the radiation output. And so they're a little complicated, but I'll walk you through it. So the y-axis, you have a % volume of an organ or the tumor volume, and you have a % dose on the x-axis. And basically what you need to know is for the tumor volume, or what we call a PTV here, you want a nice sharp corner. And that just says that your total prescription dose is covering the full tumor volume. So you're irradiating the full tumor, you're killing all the cancer cells and hopefully curing the cancer. Now your healthy organs, such as the duodenum, you want to have a curve that comes as close to the origin of the spot as possible. And that just says you're giving as little dose as possible to as little of the organ. And so in these plots, you can see a solid black line, which is what was in the initial treatment plan for the amount of dose. And the dashed line represents what was actually delivered to that patient based on these anatomical changes that happened. And if you look in this plot in particular, this is fraction 4, so this represents the 4th day of treatments. And you can see there's quite a big difference between what the intended dose was supposed to be to the duodenum and what the actual delivered dose was. And this can summate over time, and this can cause some serious side effects. Another, from the same paper, this is another plan that they did, where this air bubble in the stomach here almost tripled in size. And so because the attenuation through air is a lot less than the attenuation through tissues, what ends up happening is you end up moving the high dose region back deeper into the tissue. And so you end up missing a piece of the organ. And you can see that here, how this is a nice sharp corner with the solid black line, but the dashed line misses. And what this says is only 54% of the tumor is actually covered by the prescription dose. So we are not giving enough dose to 46% of the tumor, leaving a lot of cancer cells there, which we are not then curing the cancer. So this is obviously a problem. We don't like this. So what do we do? Well, so we're going to start actively checking for daily anatomy changes by imaging more often. So rather than just doing a single CT at the beginning, we're going to take a CT every single day. And then from there, we will check the patient anatomy and we'll re-optimize the treatment plan on the anatomy of the day. So we'll rerun that Monte Carlo simulation so that we know exactly what the dose is to each of the tumors. And this technique is called adaptive radiation therapy or radiotherapy. And this is where MR-guidance can really help us out. So MRI can be used to image the patient. So instead of using a CT, we can use an MRI instead. And there's two main benefits to this. So the MRI uses magnetic fields and an RF pulse sequence to create an image, which is not ionizing radiation. Whereas a CT uses ionizing radiation, which means that you're getting dose from that CT. So if you're taking this image more and more often, what can happen is that dose can add up over time. It could potentially lead to additional side effects that wouldn't be the case if you're only imaging once or twice throughout that course of treatment. The MRI, because you don't have the ionizing radiation, you're not giving any additional dose to the patient, which is really good. Additionally, you have much better soft tissue contrast, which gives you better visualization of different organs, especially in the abdominal region. So these two images, you have a CT on top and an MRI on the bottom. This is the same patient and the same location in the body. And so you have this big gray piece is the liver here. And I want to look at the shadow. You can see, I think the projector is okay. So this shadow, you can see it. It's somewhat clear, but it is a little hard to see, and depending on the optics of your screen, could be difficult. But then if you come down to the MRI, this big white cartoony thing is the same organ. That's the gallbladder. And so you can see how much easier it is to see and delineate how much clearer the edges are of the gallbladder simply by using an MRI instead of a CT. And that's true for other organs as well. Right here you have a piece of the bowels, which is obviously a lot clearer in the MRI than in the CT, as well as other organs throughout the abdominal region. And this is what we call MR-guided radiation therapy, or MRGRT. So just like we had the onboard imaging with the KV imagers on the traditional LINAC, we can also combine an MR and a LINAC together, what we call an MR-LINAC or an MRL. And this is a newer piece of technology within the last like 5 to 10 years. They've really come into play. And this combines the MRI and a LINAC and is to a single machine, which allows for using MRI for imaging and the treatment all in one sitting. So you don't have to put the patient in an MRI down the hallway or in a different department and then walk them down the hallway to the treatment room and get their treatment. You can do it all in one sitting, which is really nice. And then eventually this will allow for real-time imaging. So while the beam is on, you would also be able to get the image. This is not clinical yet, still in very early research stages. So online adaptive radiation therapy then is this idea where we're gonna then adapt the treatment while the patient is laying on the table, because now you have the imaging and the treatment all in one machine. So currently though, adaptation of the contours and the re-optimization can take 60 minutes or longer. And then the contouring piece, so the drawing of all the organs and outlining of all the organs, can take over 30 minutes. And this is a really long time for the patient to sit on the table, because any sort of movement of that patient can cause additional errors. And people like to wiggle. We don't like to sit still very long. So you want to be able to speed that up as fast as possible. And so the goal of online adaptive radiotherapy is then to shorten that time period and then to have totally hands-off with no manual intervention. So you don't want a doctor there having to do it all by hand. You want to use computers to do this. And this is where the development of computer processors and AI have really come into play. Which leads us into our detour into artificial intelligence land. So you have three kind of hot topic words that you'll see about, you have artificial intelligence, machine learning, and deep learning. And based on this bubble, you can see that, you know, they're all a piece of each other. So AI is at the top, machine learning is underneath that, and deep learning is underneath that. And deep learning is what I actually use in my research. So deep learning is a class of machine learning algorithms that uses multiple layers to progressively extract higher level features from the raw input. And it's originally inspired by the neural pathways in our brain, where that we're able to receive some input information and infer an outcome. And that's just through our ability to learn over time after seeing the same thing and variations of the same thing over and over and over. The difference between machine learning and deep learning is, in machine learning, the scientist first has to extract certain features that the model will then learn from. So if we're talking about image processing, that could be something like color information or texture or gradient information, edge detection. But the scientist has to choose those features and pull them out of that data before the model can learn. In deep learning, though, the scientist doesn't have to do that. The scientist just has to give the model the input data, and then from there, the model itself will choose the important features, pull them out, make those connections, and learn and be able to come up with an output. And it does require, because the model is choosing these features itself, it requires a lot more data and it's much more computationally expensive, but it does really well for applications that are much more varied and have much more variation in them and could be more complicated. So AI in medicine, just like AI in the rest of the world, is a really big topic right now. Some of the benefits include it's fast, it can be more accurate in a lot of cases, it reduces manual intervention and therefore human error that comes into play. Physician burnout is a really big problem, especially post-pandemic. So it can hopefully reduce some of that burnout and take some of the load off of the physicians, the MD doctors. And it also has a wide range of uses. But in medicine, there's some challenges that are a little bit more unique, specifically to medicine, that maybe isn't as applicable to, you know, your suggestions on Netflix, for example. So one, we have very limited data. Because we're using patient data, it's protected. There's a lot of privacy issues there. There's no public domain that we can just pull our data from. It's also very hard to label the data because you are collecting it yourself. Again, not having those public domains can be quite difficult. There is a lot of variation and some subjectivity in some cases. Additionally, if you are working with images, there can be image quality issues or imaging artifacts that occur. And most importantly, it requires very, very high accuracy. You know, back to the Netflix example, if there's a suggested movie that, you know, they think you'll like and you don't want to watch it, you just scroll on right past and call it a day and your life is no worse off. But if we're talking about like disease diagnostic, for example, or detecting a disease, if our model misses a disease, now that disease can grow and get worse and you can have some serious side effects. Or on the flip side, if we say the disease is there and it's not, now the patient has to go through a lot of extra tests that they don't necessarily need to. So the accuracy is very, very, very important in our uses. And we still need human validation because, again, the accuracy is so important that if you don't have that human validation or a lot of validation, you can't deploy anything that's not right. But there are a lot of applications that we can use AI. So the first is diagnostic medicine, specifically disease detection in images. So this is big in breast cancer detection with mammography, as well as connecting relevant symptoms for early disease detection, predicting patient outcomes earlier. In imaging, you can do image reconstruction, as well as synthetic imaging, which means converting between like CT and MRI, for example. Drug development is a certain field, and then lots of patient interactions, so scheduling, matching patient to a good physician, transcribing documents, online chatbots, etc. And then finally, in radiation therapy, specifically, we use it for online adaptive radiotherapy quite a bit, which brings us to the end of our detour. So as I mentioned, the goals of OART, or online adaptive radiotherapy, is to create this workflow that has no manual intervention and to speed it up so that it's under 30 minutes total. And deep learning is really what helps us achieve these goals. There's 3 basic steps to the workflow. You have the imaging, so taking the MRI. Then you have the contouring piece, and then finally the treatment plan optimization. And that hopefully can be done all within 30 minutes, and you have you just press a single button at the beginning, and then you end up with a treatment plan at the end that then a doctor can validate before the patient actually gets treated. And the contouring does take the longest piece, the longest amount of time, and so that's the piece that my group has focused on. The first step of this is what we call an automatic segmentation, and this is work that my colleague Dr. Amjad is working on. And basically it's a deep learning based automatic segmentation, so it's the initial attempt at getting a contour. She uses a 3D residual unit model, and what happens is you input an MRI image, and then the model takes this image and then it outputs 11 different contours for 11 different organs. And those organs are liver, spleen, kidneys, bowel, stomach, pancreas, duodenum, aorta, and esophagus. So it's a multi-organ model. It produces a lot of good results, but it's not quite good enough. So specifically the stomach is what I'm going to focus on for the rest of this talk, because it's kind of in the middle of the easy-hard spectrum. And the average DICE score, or DICE similarity coefficient for the patient, is anywhere between 0.77 and 0.95. Now the DICE score, or the DICE similarity coefficient, is represents the percent of volume overlap of the 2 contours. So if you have some ground truth contour, so what the contour is supposed to look like, and some deep learning based contour, so a contour out of this auto segmentation process. In this case, if our organ was perfectly rectangle, which is obviously not ideal, is not a real organ, but in this case it works out. You can see that this has a DICE overlap of 0.7. So you can see how poor that overlap is. And if we were to have an organ, a contour that only overlaps with a DICE of 0.7, it's not good enough for treatment. You can't use this. And what this clinically works out to be is that only about 30% of the slices are acceptable for treatment, which means 70% of the slices still require manual editing. So this doesn't work with our whole no manual editing part, no manual intervention. We still have a lot of work to do. So this is where we come up with the contour refinement workflow. So from the auto segmentation, or DLAS, you have some attempt at a contour. So you have a ground truth contour here, and then some bad deep learning contour. It'll go through a deep learning based quality assurance, though, which will say whether or not it's acceptable or requires edits. And if it requires edits, it'll go through this contour refinement. After that, it'll go through another QA, either say it's acceptable or requires editing, and then we'll keep going with our lives. And the contour refinement method is what my specific research that I work on. So as I mentioned, the automatic contour refinement can further improve the DLAS contours, these auto-segmented contours, and then continue to minimize the manual editing time that would be required and the manual intervention that would be required in this process. The benefit over the initial auto-segmentation is now we have some sort of guidance to use in our model. So from our initial MR, or our initial auto-segmentation, all you do is give it an image, and you say, okay, find the stomach. And that's a big area to look in, and you don't really know. It's kind of hard to see. But now after our segmentation, we have some sort of region of interest that we can look at now. And so we give that auto-segmented contour to our correction model, and we're able to then correct the contour so that it perfectly outlines our stomach. So the model I use is a dense UNet model, and UNet is a very common image processing and segmentation deep learning model. It's named off of the U shape that it makes. You have an encoder side, which is the side over here, and then a decoder side, which is over here. And as it goes deeper through these layers, you're consistently downsizing your image so that, and consistently looking for higher level features to connect with the output. Then as it goes up the decoder side, you're upsizing the image back to its original shape. And you have these long skip connections, which says, okay, the spatial information we found out over here now matches to the spatial information that we have over here. And that just gives that spatial information, keeps the spatial information that you wouldn't get otherwise. The dense UNet is just a little bit more sophisticated. You have these, what we call dense blocks, and those are short concatenations that just allow for feature reuse between layers. So what that means is the information that we find at these top layers is not lost by the time we get to these deeper layers. We're able to reuse the information that we already found in the higher level layers. It's set up as a 2-channel binary classification model. So each pixel in the image, it basically is gives you a probability between whether it's background or organ. So we're giving, after the image and the initial contour is going through the unit model, basically what happens is then it outputs a probability of whether or not that pixel is part of the organ or part of the background. And then when we cut off we cut off at 0.5, so 1/2, and that's what creates this binary mask and gives us the edges of that contour volume. Imaging, the image preprocessing is really important because every image has slightly different, have slight differences. And so in order for the computer to consistently learn, you need to standardize all these images. So these just include simple things like bias correction, intensity clipping, and normalization. And then we also crop closely around that deep learning segmentation contour, the initial contour. And so that just focuses the model a little bit more. That says, look at this in particular. You don't have to look at all this outside information. And then we resize all those images to the same size, to 64 by 64 pixels, and run a quick smoothing function over all of it to reduce the amount of noise in the image. So as I mentioned, I focused on the stomach contours first because it's kind of in the middle of the easy-hard spectrum. And I trained models for both the conventional MRI and the MRL images. There are slightly different image artifacts in both of them, so that's why we did 2 separate models for those. And we ended up with a patient set of 74 patients. So that's 74 image sets that we could work from, and it ends up with 1,600 slices containing the stomach contours. Those are separated in 2 categories then: acceptable, because if the contours are good, we don't have to do anything to them, so we'll just throw those out for now. And then everything else requires editing, and that set has about 1,300 slices in it. And then the set we work with for our model is split into 3 sets from there: a training set, a validation set, and a testing set. The training set is what the model actually sees during the learning process and what it's using to actually learn these features and learn how to produce an outcome. A validation test just tests generalizability, because during the training process, the model really can just memorize the data, which we know is not good enough. So you need to be able to use this on data that it hasn't been seen before. And so this validation set just kind of helps test the generalizability. And then testing set is totally independent and used to calculate our final results. 1,300 slices, though, is really not enough for training, so we do what's called data augmentation. And the augmentation just gives the model a little bit more information during the training process. We have 2 steps to this. We have an offline augmentation, which essentially creates 10 fake patients for every real patient. And so what happens is you take every single organ and slightly shift it or rotate it or move it around a little bit so it looks different from the first patient. And so you have a fake patient that could be a real patient if we had a human that it came from, but it's different from the actual real patient that we have. And then we do an online augmentation. So during training, we just, at each training iteration, we just slightly adjust every image so that it just looks a little different, because a little bit of variation can really go a long way in these models. So with all that, we end up with 15,000 to 20,000 to 20,000 slices. So 15,000, over 15,000 in our MRI model, and almost 20,000 in our MRL model. And we're testing this on 400 slices total. So in the end, our MRI model shows 64% of the slices and improved that DICE overlap score. 64% of the slices showed improved DICE overlap after the correction, and 27% of those slices became acceptable for treatment. And for the MRL model, we did even better. We had 87% of the slices show improved DICE overlap, and 31% of the slices became acceptable for treatment. So numbers are great, but let's look at some pictures. So you can see here, in these cases, the blue is the ground truth, so that's what it should look like. The green is the initial segmentation, and the red is after we ran it through our correction model. So you can see, even like in this case where the initial segmentation was kind of way off, it tried to outline something else, we were able to correct it and really get a really good overlap with what we think that with what the ground truth contour really is. And then even in these cases in the corner, you see this one here, you have the initial segmentation, try to include a piece of the spleen into the stomach contour, and our correction model was able to say that's not stomach, so we'll just remove that loop. And then on the flip side here, the initial segmentation missed a piece of the stomach, and we were able to find that with our correction model as well, which is really good. So those were MRL images. These are some MR sim, and you can see again, even where there's some pretty intense errors, we're able to correct for the contours pretty well. Now, this correction algorithm still only corrects about 30% of the slices that still required editing. So what we're left with is still 50% of the total set that requires manual editing, because you had 30% be acceptable after segmentation, 30% of the 70 set then also get acceptable. So now we're left with 50% requiring manual editing. Still not good enough. We still takes way too much time. This patient's on the table. We got to get them treated. So what our group is working on is doing this iterative contour refinement method. So basically, after the stents unit, for example, it'll go through a QA. If it's not acceptable, if it still requires editing, it'll go back here and we'll try a different method of correction. And so we'll just have this, we'll build up this set of correction methods that we can use, and it'll consistently go through this loop until we get as many of these slices corrected as possible. So some of the different methods that we're working on, we have an active contour method, which is an algebraic method. It's not an AI method. And that just looks at some gradient information. It essentially snaps the contour to the gradients around the organ. And then you have things like a basic unit model or a 2D hybrid or a mini 3D model. And those two last models basically take multiple slices, so you have 3D information now in your model that will help guide the training a little bit more. And the goal, we know that correcting every single slice is probably not like totally doable. So we're trying to get less than 5% is our realistic goal. That still require manual editing. So in conclusion, we have MRI-guided radiotherapy allows for better soft tissue visualization in the abdomen. OART allows for fast treatment plan adaptation to daily anatomical changes. Autosegmentation does not produce sufficient contours, so automatic contour refinement is necessary. And using the stents unit method that I'm working with, we're able to correct an additional 30% of the stomach contours for treatment planning. And with additional contour refinement methods, we hope to correct over 95% of the slices. And in the future, we're going to extend this to more organs. So as I mentioned, the stomach is in the middle of the easy hard spectrum. So on the harder end of the spectrum, we have things like bowels, pancreas, and duodenum that are just, there's a lot more variation in them. The stomach was all kind of, more or less uniform. It's all kind of blobby, easy shapes to look at. The bowels, which you can see two examples from the same patient. So this is just different parts of the abdomen are very, very different, and they're kind of scattered just all over. So here too, the blue is the ground truth, and then the green is the initial autosegmentation. And so in the case here, you have, a little bowel here, a little bowel here, a little bit here, 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 here. They're just kind of scattered all over. And the segmentation just tries to outline everything, right? It includes the sections here, you have some extra loops here. And so it's really hard to correct them because you have the guidance that comes from that initial segmentation is not very good. On the other hand, again, on this side, now you have the segmentation that's totally missing a part of the bowel. And so now our correction model has to find this piece that it was missing initially. And so these just create like different complications. On the other end of the spectrum, you have easier organs to deal with. You have things like liver, kidney, aorta, and esophagus. And those are really easy to see. They're really solid masses. So in this image here, this is the kidney. So you can see how easy it is to find. But what that challenge then is for the correction method is the initial autosegmentation is very, very good, which is great, right? That's what we want. But that means that there's less training data for us to work with for these correction models. And then that just can cause challenges when it comes to training models for our purposes. And so lastly, I'd like to acknowledge all of my collaborators. This is a really, really big project that I work on one piece of, and it really is a big group effort that we're working on. So my advisors, Drs. Lee, Zhang, and Paulson at MCW have been really influential, as well as the entire MCW auto segmentation group. Doctors Amjad and Dr. Conlon, they are in charge of creating all of our ground truth contours and creating all of our training data, as well have been working on this like initial auto segmentation method. Doctors Ding and Dr. Zarania, they are working on other correction methods with me. And then Dr. Ding worked on the augmentation, is also doing that QA piece that I briefly mentioned. Lastly, the MCW and Frater Health MRL team, and then my funding support through NIH and MCW. My references, yeah, thank you. And I think we have time for questions, probably. Yeah.
What is the duodenum?
The duodenum?
The duodenum.
So it's part of the gastrointestinal tract, and it helps break down food, essentially. Yeah.
You were talking about how the patients have to wiggle a lot.
Mm-hmm.
That's not programmed like a move with the wiggle, or does it?
So that is one of the things that I mentioned, this online imaging. So you could do the imaging during the treatment, and that would the goal with that is so that you would be able to monitor all the wiggles during the actual treatment. Right now it's difficult because it takes so long to do this adaptation that you can't, if you image, adapt the plan, and they wiggle during that, so you re-image, and then you have to readapt the plan, and so it would be this like endless cycle. So right now we have right now how we deal with it is we just add in extra margins around the tumor, so you are not getting as conformal of a dose distribution as you would want. You just add in, you know that the patient's going to move, so you just add in that extra space around the tumor to make sure that you're covering everything you need to. Mm-hmm. Yeah.
So since there's no like public domain with all this data,
Yeah.
like, that makes the job a lot harder.
Yeah. Yeah, so the MRL, so we were one of the first in the country. The Medical College of Wisconsin was one of the first in the country to get one of these for research purposes, and we've had it for about 5 years, and we have 74 patients that we can use data from. So that tells you just kind of how long it's been taking to like collect all of this data for us to use. If you were using like CT data, for example, that because that's been clinical for a lot longer, there's a little bit more of a backlog you could use, but especially when you're using like these new imaging modalities, it's a lot harder to collect all that data. Mm-hmm. Yeah.
So I know a person who was doing
Yeah.
to read zip codes.
Okay.
Right.
Sure.
And their goal was always to get the algorithm to do better than humans.
Yeah.
So if you went back in your flow there,
Mm-hmm.
if you replace the corrector with a different doctor,
Yeah.
how many would a second doctor effectively get?
Yeah. So that's the question we're still looking at. So there has been a lot of studies. So because currently all of these like contours are so manual, there's variations that go up to like 5% of volume. And the hard thing is these variations in the contours do not make any dosimetric input difference. So the dose is the same to the tumor regardless of how the doctors draw it. But because that's the case, a doctor goes, well, mine's right, and this other doctor goes, well, mine's right, and I'm not going to change it. And so when you have doctors evaluating these contours, you will get different answers. And so that's there's a couple different ways that you can like deal with that. The first is to just say, well, we're just going to have one person do it and everyone else needs to deal with it. Or there's been ways to do physician-specific networks where you train a general model and then from there, you're able to train networks specific to each physician. Or you can just train that general model and say, like, is this good enough? We know it might not be perfect, but good enough is okay in this case. So yeah, that's one of the pushbacks too from actually implementing this into the clinic. Yeah, I know.
A lot of zip codes.
Yeah, it's a lot easier. That would be ideal.
Do you take volunteers to do scans?
I don't know. Probably not. Not for our purposes because it's you have to get the radiation with it, and you can't ethically give radiation to someone who doesn't need it. So even so that's the main issue with our purposes. But I know there are like clinical trials, like through the hospital that request MRI volunteers all the time because that has no issues for the patients. So I don't know. Yeah, but not for us, unfortunately.
I just have one.
Yeah.
So for all the people out there that do radiography,
Yeah.
it's not a bad thing if it's not good.
It teaches you resilience. 4 years. So I spent 4 years on the car paint. Nothing worked. We redid the analysis, I think every year pretty much for various reasons. But yeah, it definitely teaches you resilience, and like even because with this deep learning stuff, it's a lot of trial and error finding the right parameters to set up your model correctly. And so I'm in one right now I'm working on one model right now that hasn't been working for a while, and you just got to keep pushing on, and that's okay because then when it does work, it's even more exciting, and you get really, really excited.
All right, let's give her one more round of applause.